Hello, good evening, dear students. Last class we were discussing about biomolecules, right? So let us just recap, okay, what all things we have discussed about biomolecules. So, what are biomolecules? What did we discuss about biomolecules, students? What are biomolecules? Yes. What do you mean by biomolecules? So, these are the complex organic substance, right? So, with the help of these biomolecules only, the living organism, okay, so you can build up the living organism. And it is required for what? It is required for their growth and maintenance, right? So, which are the different biomolecules? Carbohydrate, proteins, nucleic acids, lipids, right? So, these are all the biomolecules. Then we have discussed about something called as biochemistry. What do you mean by biochemistry? So it is the branch of chemistry which deals with what? It deals with the structure, functions, and the interactions of these biomolecules. Okay, they are called as the biochemistry. Then we have discussed about the so these things we have discussed, right? So then we have discussed, so or we are going to discuss about carbohydrate, right? Proteins, what are vitamins, nucleic acids, enzymes. So these things we are discussing, right? So we have already discussed about carbohydrates. Okay. So these are the main, what you can say, biomolecules. What are the building block and what are the major functions? So proteins, basically they are made up of the amino acids. Okay. And that is the basic structure and function of the cell. Then DNA, it is called as a deoxyribonucleotide. Okay, so it is a hereditary information. It gives you the hereditary information. Then what do you mean by RNA? It is ribonucleotide. So it helps in the protein synthesis. Okay, so the function of RNA is protein synthesis. Then polysaccharide. So it is a what it is made up of monosaccharide units. So when this monosaccharide units, they are in many numbers. Okay, so they make the polysaccharide, right? So these are the storage form of energy. They are the storage form of energy. Okay. Then lipids. What are lipids? They are the fatty acids and glycerol. So they are the made up of fatty acids and glycerol. So they are nothing but the storage form of energy. Okay, to meet the long term demand. Okay, so these are some things. Okay, so then we have discussed about the carbohydrates, right? So we have discussed about the carbohydrates. So let us go stepwise. Okay, so we are discussed about carbohydrates. Okay, what are carbohydrates? They are considered to be the hydrates of carbon, right? With the general formula CnH2n, right? Then we have seen the different examples of carbohydrate, glucose, fructose, sucrose, right? Yes, then we have discussed about the classification of carbohydrates. So based on how many number of simple units are given out. So monosaccharide means one. Okay, so these are the simplest carbohydrates which cannot undergo hydrolysis, right? Which examples we have given? Which examples we have given? We glucose, fructose, right? So these are the examples of the monosaccharide. Then oligosaccharide means the carbohydrates which on hydrolysis gives you 2 to 10 monosaccharide unit, right? So here also, then we have discussed about these oligosaccharides. Further, they are classified as disaccharide, trisaccharides, and tetra. Di means 2, tri means 3, tetra means 4. Okay, based on further hydrolysis, they will give you so many this what units of the monosaccharide units, right? Okay, then poly means the carbohydrate which on hydrolysis give you large number of monosaccharide units. So example is starch, cellulose, glycogen, etc. Then also we have discussed about something called as reducing sugars, non-reducing sugars, right? Reducing means what? The carbohydrates which will reduce the Fehling solutions, okay? And tolerance reagent. And non-reducing means the carbohydrates which do not reduce the Fehling solution and tolerance reagent, okay? So what is example of non-reducing? It is sucrose. Then we have discussed about what are monosaccharides. We have seen the example of glucose, right? Then we have seen the structural elucidation of glucose. So all the reactions are very important. Okay, the students who have not were not present for the last class, you can go through the video. Okay, in the last class we have already discussed about these things. Okay, so the structure elucidation of glucose, how we get the structure. Okay, six carbon structure, six carbon structure of the glucose. Okay, so these things we have discussed. Then we have discussed what the cyclic structure of glucose. Okay, how to write the hemiacetal structures, then the Hayward structure. Very important. Please practice all the Hayward structure. Okay, for glucose, fructose, maltose. Okay, then your sucrose. Okay, lactose. So if all the Hayward structure, please do practice. Then we have studied about fructose. Then we have studied about the disaccharide example maltose, which is called as a malt sugar. Then also we have discussed about sucrose and lactose. Lactose is the milk sugar. So these all have our structures. Please practice. Then polysaccharide. What do you mean by polysaccharide? We have discussed. Then the example starch. Okay. Cellulose, glycogens. Okay. Then we have seen the importance of the what carbohydrate. So these all things we have already discussed. Okay, in the last class. So now then we have discussed about the something called as the proteins. Okay, then we have discussed about what proteins. So proteins, as we know, so they are basically made up of the amino acids, right? So amino acids. So the amino acids are the building block of this protein. Okay, so in a what is amino acid? So they are the compound which consists of a amino group and a carboxylic acid group. Okay, in the same molecule, you can see those structures, right? So yes, you can see the amino acid structure. So you can see that there is one amino group and there is one carboxylic acid group, right? Yes. So this also things we have discussed. Then we know that there are 20 naturally occurring amino acids. Okay. Glycine, aniline, valine, okay, leucine, isoleucine, arginine, then lysine, glutamic acid, aspartic acid, glutamine. Okay. Then you have threonine, asparagine, okay, then serine, cysteine is there, methionine is there, phenylalanine, tyrosine, tryptophan, histidine, okay. Then you have proline. Okay, so these are all the 20 amino acids. So here you can see all the 20 amino acids. Okay, based. So they are all the naturally occurring amino acids. So these are again further classified as neutral, acidic, okay, and basic. Acidic means what? The number of carboxylic acids are more, okay, more than one. And basic means the number of amino acid is more, okay, more than one. Then they are basic. Neutral means same number of carboxylic acid and amino acid. Okay, so these are all examples you can go through. Then there is something called as sulfur containing amino acid, alcoholic, okay, which has alcoholic group, aromatic amino acid and heterocyclic. So we have classified them usually. Okay, then we have also studied about what are essential and non-essential. Essential amino acids are those which cannot be synthesized in our body and has to be taken through diet. Okay, so the food which we ate, through that food we are going to eat, we are going to take this essential amino acid. And non-essential means what? Non-essential means they are synthesized in our body. Okay, they are synthesized in our body. Okay, then we have seen the what peptide and polypeptide, right? Peptide means what? Peptide formation. So peptide means the amide bond, okay, which is formed between a amino acid, between the two amino acids with the elimination of water molecule, okay. So that is a peptide bond. So you can see this is a peptide linkage, CONH. Okay. Then what is a polypeptide? Okay, polypeptide means more. So suppose if I want to say dipeptide, so dipeptide how it is formed? When two amino acids they are linked, okay, you get one peptide bond that is called as a what? So like that. So then so depending upon the number of peptide bond in di, tri, tetra, and penta. Okay, so like in di you have one. In tri, you have two peptide bonds. In tetra, you have three. And penta, you have four, respectively. What is a polypeptide then? So when more than 10 amino acids, they undergo condensation. Okay, the product formed is called as a polypeptide and it will have molar mass less than 10,000 unified mass. Okay, then we have discussed about the proteins. Okay, what are proteins? Then, so what are proteins? They are the polypeptide. Okay, when a polypeptide is more than 100 amino acids, okay, that is their mass is more than the 10,000 unified mass, then it is called as protein. So again, we have seen proteins. Okay, fibrous proteins. What are globular proteins? Fibrous means they are in the long parallel polypeptide chain. Okay, they are like thread. So what we can give example, the keratin, hair. Okay, it is present in hair. Then myosin, present in your muscles and wool. Okay, it is present in silk. Then globular proteins means these are coiled shape. Okay, these are also the polypeptide chain, but they are in the coiled form. Okay, they'll give you a spherical shape. And these are usually soluble in water. For example, your albumin and insulin. Okay, then we have seen the structure of protein, that is primary structure, secondary structure, tertiary structure, and quaternary structure. So these all structures also we have discussed in the last class, right? So so today we are going to start with the next part, that is enzymes. Okay, next we are going to discuss about enzymes. Okay. So, so these are the things which we have discussed in our last class. So today let us see further. So what are enzymes? So we are going to discuss about what are enzymes. So what are enzymes now? So enzymes are they are the globular proteins. Okay, they are what? They are the globular proteins. Okay, they are the globular proteins which are produced by what? They are produced by living organism. Okay, they are produced by living organism. Okay, they are produced by living organism. Okay, so that it can catalyze some biochemical reaction. Okay, now, these are very specific in the action. Okay, they are very specific in the action, enzymes. Okay, and so there are different enzymes. Okay, there are different enzymes. So the this efficiency of this enzyme, it depends on the temperature and the pH of the solution. What is the efficiency? It depends on what? Two factors, temperature and pH. Okay. The optimum efficiency of this enzyme, okay, it is at 37 degrees Celsius. Okay. So maltose. So maltose, what is present in maltose? So maltose in the when you use the enzyme this maltase. Okay, maltase is the enzyme which will break down the maltose into 2 glucose unit. Okay, to 2 glucose unit. Uh, and then there is an enzyme called lipase. Okay, so it is used in hydrolysis of what? It is used in hydrolysis of oils and what? Oils and fats. Okay, it is used in the hydrolysis of oils and fats. Then your enzyme. So there is one enzyme which is present in saliva. Okay, in the in the saliva, what it does, it decomposes. Okay, decomposes starch. It decomposes starch into maltose. Okay, it decomposes starch into maltose. Fine. Yes. So and these are the things which you are going to study in enzyme. Okay. So enzymes, what they are the globular proteins which are produced by living organism. Okay, so that it can catalyze some specific biochemical reaction. So on what factor the enzyme depend on? They are, as you know, enzymes are very specific in action. Okay, so it depends on the temperature and pH. So the optimum temperature is 37 degrees Celsius. Okay, so maltose, when it breaks down to glucose, so which is the enzyme used? Maltase. Okay, then lipase, enzyme lipase, it is used in hydrolysis of oils and fats. Okay, and enzyme which is present in saliva, it decomposes starch into maltose. So these are the things which you have to remember under enzymes. Is it clear? Okay. So next we are going to discuss about vitamins. Okay, next we are going to discuss about vitamins. Now, what are these vitamins? Is it necessary for is the vitamins necessary for our body? Yes, are they necessary for our body? Yes, right. So these are also the organic compound, right? These are the organic compound, okay, which are required in food. So the food which we are eating, it should have all these biomolecules, right? So we have to consume all this, what? Biomolecules. So vitamins, so these are the organic compounds which are required in diet in very small quantity. Okay, in small quantity we require them. Okay, so that it can perform some. So when we are having this, okay, so what it will do, it will perform, okay, some specific. So it performs specific biological functions. Okay, it gives you some, it will, so because of the presence of the vitamins, it plays a major role in our living being system. Okay, so so what it does, there should be so proper maintenance, okay, and growth. So these vitamins, they help in the proper maintenance and growth, okay, and also the health of an organism, okay, health of an organism. So, vitamins are necessary. Yes, vitamins are necessary. Why they are necessary? Because they are the organic compound which are required in our diet. Okay, which in small quantity, and it helps to perform the specific biological functions. Okay, for the maintenance and growth. Okay, and also the health of an organism. Okay, so this is about the vitamins. Next is classification. Okay, what are the classification of this vitamins? Okay, so this vitamins, it depends on the solubility. Okay, it depends on the solubility. Solubility. Okay, whether it is soluble in fat or it is soluble in water. So they are classified as two types. So whether they are soluble in fat or water. So they are called as a fat soluble vitamins. Okay, fat soluble vitamins, and second one is the what? Water soluble vitamins. Okay, water soluble vitamins. Can you answer what is the meaning of fat soluble and what is the meaning of water soluble? Fat soluble means what? These vitamins, they are soluble in fat. Okay, these vitamins are soluble in fats and oils. Okay, they are soluble, but they are insoluble in water. Okay, but they are insoluble in water. So you need to remember the example. Okay, where they are stored, then they are stored in liver. Okay, they are stored in liver and adipose tissue. Okay, it is stored in the liver and adipose tissue. Okay, which is the example? So the example of fat, these are vitamin A, D, E, and K. Okay, vitamin A, D, E, and K. Then what are the water soluble? So these are soluble in water. Okay, these are soluble in water. Okay, and they are supplied regularly in diet. Okay, they are supplied regularly in diet. Okay. These are excreted. Okay, these are given out. These vitamins are given out and they cannot be stored in water. Okay, they cannot be stored in body. They cannot be stored in body. They are given out. So these are soluble in water. They are supplied regularly in the diet. Okay, and they cannot be stored in the body. So what is the examples? Vitamin B group. Okay, and vitamin C. Except your vitamin B12. Okay, all other are water soluble vitamin. Okay, vitamin B group and vitamin C. So all these are what? They are the water soluble. So remember what are these vitamins. So how they are classified based on the whether solubility. Okay, whether they are soluble in water or fat. So fat soluble and water soluble. Is it clear? Okay. Now we are going to see some of the vitamins. Okay, they are this what due to the deficiency of which this what your due to the deficiency of which what you can call vitamin. Okay, what disease is caused? Okay, what disease is caused? So that we will see now. So when you have vitamin A, so when there is vitamin A deficiency, okay, so that is it will cause loss of vision. Okay, it will cause loss of vision. So blindness. Okay, blindness is caused due to the vitamin A, that is retinol. Okay, that is retinol. Then vitamin B1. Okay, that is due to thiamine. Okay, due to thiamine. So what happens? There will be beriberi. So the disease caused is beriberi. That is what you can. So the muscles and okay, muscles becomes weak and there will be severe weight loss. Okay, there will be severe weight loss. The muscles become weak and there is severe weight loss. Okay. Then vitamin B2. Vitamin B2, that is riboflavin. Okay, riboflavin. So what it is used? So it is ariboflavilosis. Okay, it is ariboflavinosis. Okay, that means due to poor diet, okay, deficiency disorder, poor diet and mouth sores. You get mouth sore. Okay, so that is vitamin B2. Then vitamin B3. What is vitamin B3? Niacin. Okay, niacin. That is pellagra. Okay, pellagra. That means diarrhea and dermatitis. Okay. Then vitamin B6. Vitamin B6. That is pantothenic acid. Okay, pantothenic acid. That is uh, paresthesia. Okay, it is paresthesia. Okay, what happens here? Okay, there is attacks and multiple, okay, it affects your brain. Okay, so there will be effect at your brain. Next is vitamin B7. Okay, that is biotin. Okay, that is biotin. So you get dermatitis. Okay, you get dermatitis. Okay, that is red and itchy skin. So your skin becomes red and you will get itchiness. Okay, then is vitamin B9. Okay, vitamin B9 is folic acid. Okay, folic acid. So there will be what immature RBCs. Okay, you will have immature RBCs. Okay, and in your bone marrow. Okay, in the bone marrow there will be what immature RBCs. Then vitamin B12. Vitamin B12, that is cyanocobalamin. Okay, cyanocobalamin. Okay, so that is you will have your pernicious anemia. Okay, pernicious anemia. Okay, anemia. So what will happen here? So there will be an abnormal absorption in the small intestine. Okay, so pernicious anemia. Then vitamin C. So what is vitamin C? That is a ascorbic acid. Okay, ascorbic acid. So you will have scurvy. Okay, you have scurvy. That is your gums. Okay, and the joints will be swollen. So your bleeding gums and swollen joints. Okay. Then vitamin D. What is vitamin D? Vitamin D means so you will get rickets. Vitamin D is rickets. Okay, bone will become at the joints. Okay, it will become weak. Then vitamin E. What is vitamin E? So vitamin E is your hemolytic anemia. Okay, hemolytic anemia. Okay, so what happens here? Faster depletion of RBCs from the body. So from your body, the RBCs will get, it becomes less. Okay, then vitamin K. Vitamin K, what happens in vitamin K? You will have bleeding. Okay, that is, you will have irregular as well as slow bleeding and blood clotting will be there. Okay, it is called as bleeding diathesis. Okay, diathesis. So this is about your, okay. So these are the, So these are what? These are the vitamins, okay, and the deficiency. Okay, and these are the deficiencies and their disorder. Okay, so you can see. So these are all. So any they'll ask in the exam. So vitamin A causes what? A, beriberi, pellagra. Okay, then C, night blindness. D, scurvy. So you have to write the correct answer, and so you have to mark the correct answer. So like this, the questions will be. So you should know which vitamin have which deficiency. Okay, which vitamins, okay, which vitamin will cause which deficiency. Is it clear? Okay, so what is so these all things you have to know. Okay, so next we are going to discuss about nucleic acid. Okay, next what we are going to discuss, we are going to discuss about nucleic acid. So when you're discussing about nucleic acid, okay, so the what is the composition of this nucleic acid? Okay, so when this nucleic acid, when it undergoes hydrolysis, it will break down. Okay, so which are the nucleic acid? That is DNA and RNA. Okay, so when this nucleic acid will break down, this nucleic acid it will break down into what? So nucleic acid it will break down with the help of a nuclease. Okay, the enzyme is nuclease. It breaks down into nucleotides. Okay, it breaks down into nucleotides. These nucleotides further will break down into nucleosides. Okay, nucleosides plus phosphate. Okay, then this nucleoside in the presence of nucleosidase. Okay, the enzyme is nucleosidase. Okay, what you are going to get? You are going to get pentose sugar. Okay, you are going to get pentose sugar plus nitrogen base. Okay, plus nitrogen base. So nucleic acid, what are the compositions? So when it undergoes hydrolysis, okay, so nucleic acid is mainly your RNA and DNA. Okay, so when they undergo breakdown, okay, when they undergo hydrolysis, it will break down into different units. So with the help of nuclease, it will break down into nucleotides. In the help of nucleotide enzyme, you are going to get nucleoside plus phosphate. And in the presence of nucleosidase, nucleoside is further going to break down into pentose sugar plus nitrogen base. So chemically, so what does the nucleic acids are made up of? They are made up of the pentose sugar. Okay, they are made up of what? Pentose sugar. They are made up of phosphate, and they are made up of nitrogen containing heterocyclic compounds. Is it clear? So what are nucleic acid? Nucleic acid are basically they are made up of phosphate, pentose sugar, and nitrogen containing heterogeneous compound. Okay, so these are called as a long chain polymer of nucleotides. Okay, they are defined as a long chain polymers of nucleotides. Okay, that is your polynucleotide. Now let us understand what are one thing. Okay, what is a pentose sugar? Okay, what is the so this we will understand. So what is a pentose sugar? So these all things we will discuss. Okay. Now pentose sugar. First we are discussing about pentose sugar. So in RNA, so we have discussed about two nucleic acids. One is RNA, another is DNA. Okay. Now another is DNA. Okay. So in RNA, okay, the sugar which is present is what? It is the beta D ribose. Okay, it is beta D ribose. Whereas in DNA which is present, it is beta D 2 deoxyribose. Okay, so in DNA, which is the sugar which is present, it is your beta D 2 deoxyribose, and in RNA, which is a sugar which is present, it is beta D ribose. Is it clear? Okay, so pentose sugars in RNA, the sugar present is beta D ribose, whereas in DNA, it is the beta D 2 deoxyribose. Please study the structure. Okay, structures are very, very important. Okay, structures are very, very important. So what you can see, okay, both are the what 5-membered ring. Okay, here you can see both is hydrogen. Okay, here it is oxygen. So this is the only change. Okay, this is the only change. At second, you have what? You have there is no oxygen group, so it is deoxyribose. Here it is a OH group is present. Okay, so the sugar present are deoxyribose. Okay, deoxyribose and ribose. Now let us see nitrogenous bases. Okay, nitrogenous bases. So basically the DNA, it consists of four. Okay, four bases. It consists of four nitrogenous bases. That is adenine. Okay, that is adenine. Okay, then adenine. Then you have guanine. Okay, then you have guanine. Then you have cytosine. Then you have cytosine, then you have thymine. So these are the what nitrogenous bases which are present in what? DNA. Okay. And in RNA, what is present? Let me mark it with another color. In RNA, what is present? So the first three bases are same. 1, 2, and 3 is same. But instead of thymine, what you have? Instead of thymine, what you have? The it is uracil. Okay, it is uracil. So in RNA, you have uracil instead of thymine. So all these structures are also very important. So which are the four bases which is present in DNA? It is adenine, thymine, guanine, and cytosine. Okay, and which is the this basis present in your RNA? Yes, which are the bases? It is adenine, then your guanine, okay, then your cytosine and uracil. Okay, so let me write it properly so that you will come to know. So A, G, C, and T. So instead of thymine, you have guanine, uracil. Okay, so all these structures are very important. Now, adenine and guanine, they are the purines. Okay, they are called as purine. And cytosine, uracil, and thymine, they are called as a pyrimidine. Okay, yes. Next, let us see the structure of nucleic acid. Okay, next let us see the structure of nucleic acid. Okay, nucleic acid. Okay, so a unit which is formed by the attachment of a base. So there should be a base present. Okay, to a to the first position of sugar is called as the nucleotide. Okay, it is called as what? Nucleotide. Okay, and this nucleoside is nucleoside. Okay, and when this nucleoside, it is attached to a phosphate group at fifth position. Okay, then it is called as a nucleotide. So you can see these are the various nucleosides. Okay, so these are the various nucleosides. So these are all your nucleotide. So what you can see here, so there is one base. Okay, you have one sugar here, there is one base and a phosphate. Okay, so these are all nucleotides. Okay, so this is deoxyadenosine, this is deoxyguanosine, thymidine, and cytidine. So what you can see, so nucleotides, how they are joined? They are joined by what? They are joined together by a linkage which is called as the phosphodiester linkage. Okay, which is present at 5' and 3' carbon atoms. Okay, which is present at 5' and 3' carbon atoms of these pentose sugar. So how the nucleotides are joined? They are joined together by the phosphodiester linkages. Okay, so phosphodiester linkage is present in this your what in your so here you can see. Okay, here you can see. Okay, so what you can see, so there is a base. So how does a nucleotide is made? It is made up of this phosphate group, one sugar and one nitrogenous base. And how it is linked? How it is linked? It is linked through a phosphodiester linkage. Okay, it is linked to a phosphodiester linkage. Okay, so base is connected to sugar. Okay, base is connected to sugar and sugar is connected to phosphate. Again, this linkage, it goes on. So again, the phosphate, it is connected to sugar, okay, base. again phosphate, and the connection goes on. So this is a phosphodiester linkage which is present. Now let us see the structure of DNA and RNA. Okay, let us see the structure of DNA and RNA. So yes, so DNA is like this. Okay, it is a double stranded. Okay, like your ladder. Okay, like the ladder. So how is the ladder at our home? Okay, everybody might have seen the ladder. So this is the ladder. But here, what is happening? It is in the what? It is in the coiled form. Okay, this here the ladder is in the coiled form. Okay, so it is a double. So like this. So it is in the double. So DNA has a double-stranded helix structure, and this was proposed by Watson and Crick. Okay, Watson and Crick, they proposed a structure. So these two nucleic acids, they are held together by hydrogen bond. So the bond which is present here, okay, the bond which is present between these two, nucleic acid chain, they are what? Okay, so the bond which is present, they are the hydrogen bond. Here you can see, right? So here you can see. So this is the, so these are the two chains. Okay, there are two chains of this nucleic acid, and how they are joined? They are joined by the hydrogen bond. Okay, between the pairs of this bases. Always A will join with what? A will join with your adenine will G C and A thymine. Okay. So adenine will go on this, and thymine guanine will have a triple bond between them. So A, so in DNA it will be A double bond T. So between the adenine and guanine pair, double bond is present. Okay, hydrogen, and between guanine and cytosine, triple bond is present. So you can see the pairing here, right? So the two strands, they are com complementary to each other. Okay, now here if it is 3 dash and 5 dash, so here it will be 3 dash and 5 dash like this. So it is complementary to each other. Okay, and because the hydrogen bonds, they are formed between the specific pairs of bases. So there is, so adenine, it forms hydrogen bond with thymine, and cytosine forms hydrogen bond with guanine. Okay, so this is the structure. And when you see the RNA, it is a single stranded. What it is a single stranded. So here you can see the DNA. Okay, you can see DNA. So one is your 5 dash end, and here it is. So you can see this is 5 dash and this is 3 dash. This is 5 dash, this is 3 dash. So, and how the bases are arranged, they are, so they are in, so like this, the bases will be present. Between these bases, hydrogen bond is present. So A will pair with your T, and G will pair with your C. Okay, always this only. Okay, and RNA is what? It is a single stranded. Okay, it is single stranded. Here you can see RNA, how it is. Okay, it is a single stranded. Okay, it is a polynucleotide chain. Okay, so this will further give you to mRNA, rRNA, and tRNA. Okay, messenger RNA. Okay, then so these are the what? Different RNA. Okay, RNA is a single stranded of the polynucleotide chain that is made up of mRNA, rRNA, and the tRNA. Understood? Yes. So what is the biological importance of the of this nucleic acid? So DNA, it is the it reserves your genetic information. It tells you about the genetic information. Okay, DNA tells you what? It tells you about the genetic information. Then DNA, what it tells, it will control the synthesis of RNA. Okay, and the various proteins. So it controls. Okay, it controls the synthesis of the RNA and various proteins. Okay, then when you say the DNA, so what DNA does, it does the it always replicate itself. Okay, and because of this, as it replicates, it is the main reason, okay, for the reproduction of plants and animals. Okay, so basically, so this is about the DNA and RNA. Okay, next small concept is hormones. Okay, next small concept is what about hormones. Now, what do you mean by hormones, student? Yes, what do you mean by hormones? These are the proteins. Okay, let me write. So, what are these hormones? So, hormones, these are the proteins. So, these are the proteins. Okay, secreted. These are the proteins secreted by ductless glands of the body. Okay, of the body. So these are the proteins which are secreted by ductless glands of the body. Okay, so the hormones, these are secreted by the glands. What they do, they enter the blood. Okay, they enter into the blood. Okay, okay, they first enter into the blood and then into a particular tissue, and then it goes to a particular tissue. Okay, it then goes to a particular issue so that it can initiate a specific activity. Okay, it can initiate a specific. Okay, it can initiate a specific activity. Understood? So hormones, they are the proteins which are secreted by ductless gland of the body. Okay, and they enter into the blood. Okay, when they are secreted by the glands, they enter into the blood and then go into a particular tissue to initiate the specific activity. Now, what are the different types? So polypeptides. Okay, polypeptides. So which are the different types of examples of polypeptide? One is insulin, another is glycogen. Okay, so what it does, if there is a glycogen increase, okay, it will maintain the blood sugar level. Okay, what it does, it maintains the sugar level in blood. It maintains the sugar level in blood. Okay, so then amino acid derivative. Okay, epiferrin and tyrosine. Epiferrin, it brings out the response to stimuli. Okay, so whatever the response will be there, for that you will react, right? So that will be because of the epiferrin. Okay, and thyroxine, it helps in growth and development. Then steroids, so there are two steroids, that is estrogen and androgen. So these are the development of secondary sexual characters. Okay, so the functions of these, so these are different types, examples and functions, okay, of the different hormones. Is it clear? Okay, so what are hormones? Yes, what are hormones basically? So these are the proteins which are secreted by what? Which is secreted by ductless glands, okay, of the body. Okay, and they enter into what? So how they are secreted? So you know they are secreted by ductless gland, and slowly they enter into the blood and then at a particular tissue so that it can initiate a specific activity. Okay, is it clear? Yes. So what all things we have discussed? So this completes your chapter biomolecules. Okay. So what all things we have discussed in this chapter? We have discussed what are biomolecules, right? Then we have discussed about the different biomolecules, right? We have discussed about the different biomolecules. So we have discussed in detail about carbohydrates, then proteins, amino acids. Okay, we have discussed about enzymes. Okay, nucleotides, vitamins. Okay, hormones. So these all things we have discussed in detail. So these are the different biomolecule. Any doubt? Yes. If you have any doubts, you can ask. Use the chat box for asking a question. Yes. Okay. So no doubts. So yes, so this completes your chapters. Okay, so we have completed all the organic chapters, that is haloalkanes, haloarenes, alcohol, phenols and ethers, aldehyde, ketones and carboxylic acid, amines, that is the compounds containing nitrogen atom, and biomolecules. So these are all the organic chapters. Okay, so we have completed all these chapters. Okay, so any doubts up to here? You can, if you have any doubts, please ask. Yes. Okay, fine. Okay, so there are no doubts. Okay, fine. So I shall end the session here. So thank you, dear students.